Hello, in this lecture I will give you an introduction to oxidative stress, or more specifically the redox balance, which is really important for all organisms to deal with stress. A short definition of oxidative stress is the imbalance between prooxidants on one hand side and antioxidants on the other hand, in favor of the former. And a good example of these prooxidants are reactive oxygen species, or ROS, that can lead to cellular damage and eventually cell death, but that can also contribute to signal transduction. But where does this oxygen come from? And for that we have to go to the formation of Earth from its beginning. And we can see that 2.2 billion years ago, we had a great oxidation event when oxygen was entering into the atmosphere, and later on we have a second event in which we are currently living, in which we had a second major rise of atmospheric oxygen. And at the moment it's mainly plants that through photosynthesis produce oxygen on Earth. And you can see here that the outcome of the reaction of photosynthesis is oxygen as a byproduct. But oxygen is of course essential for all aerobic life. If we can see here, and we look into the cellular respiration, in which we produce energy in our mitochondria, then we can see that for the final electron complex in the electron transfer chain, oxygen is needed, that we are able to convert actually ADP into ATP that we can use as energy. But besides the cellular respiration, we have several oxygenases and oxidases that really use oxygen as a substrate to perform their reactions. If we look into the system or the real molecular structure of oxygen, then we can see that it's a very unique molecule, because in its ground state, oxygen has two unpaired electrons with a parallel spin. This means that the electrons cannot be shared in a covalent bond, and hence it's a biradical. So it has two free electrons, as you can see here on this slide. But of course we can make reactive oxygen species from this molecular oxygen through two processes. The first one is energy transfer. By giving extra energy to this ground state molecular oxygen, we convert one electron spin in the opposite way, and then we can have a spontaneous production of singlet oxygen, where we don't have a radical structure anymore, and where the electrons are shared in a bond. But this singlet oxygen is more reactive than the molecular oxygen. A second way in which reactive oxygen species can be formed from molecular oxygen is via electron transfer. Giving an electron extra to oxygen will lead to the production of superoxide radical, which is negatively loaded, but with one free electron, so highly reactive. Giving extra electrons and protons will convert this molecule to hydrogen peroxide. Hydrogen peroxide, which is not a radical, so this is really important for future goal of this molecule and in regulating cellular responses. Adding up an extra electron will then give rise to the very, very reactive hydroxyl free radical. And finally, an extra electron will lead to water. So in a 4 steps electron reduction, oxygen is reduced to water. Very important to mention is that also redox active metals like copper and iron can add up to the production of reactive oxygen species, as you can see here by the Fenton and Haber-Weiss reaction. In this table, some examples of reactive oxygen species are mentioned. So we have mentioned already the superoxide and hydroxyl, and the hydrogen peroxide, which is a non-radical. But of course, if they interact with organic molecules or with others, we can see that we have different kinds of radicals that belong to the prooxidants and non-radicals. But where are these reactive oxygen species produced? We can find them back in different subcellular compartments, like for instance peroxisomes, mitochondria, the endoplasmatic reticulum, and the plasma membrane. Let's dive into these different compartments. In the peroxisomes, lots of molecules, metabolites are oxidized, for instance fatty acids and amino acids, and in this process we have the transfer to oxygen with the production of hydrogen peroxide. If we look into the mitochondria, as mentioned before, we need oxygen because we have the cellular respiration. But if we then dive into the electron transport chain, electrons are transferred from one complex to another, and we face leakages at certain complexes to oxygen, leading to the production of superoxide. If we look at the metabolic activities of the endoplasmic reticulum, or ER, then we see that oxidative protein folding is very important. And in this process, you can see that hydrogen peroxide is a byproduct. On the other hand, cytochrome P450 metabolism is very important, and in this metabolism we try to convert toxic compounds to a more polar structure that we can eliminate, and in this way the cytochrome P450 metabolism is a crucial part of the ATME in toxicokinetics. ATME, which means absorption, distribution, metabolism, and elimination. And metabolism, there is where the cytochrome P450 is very important. And as is highlighted here, superoxide is or can be a byproduct of this metabolism. And then also NADPH oxidases are located at the ER membrane and can in this way produce reactive oxygen species. And if we then look at these NADPH oxidases, or we call them also NOx in humans, what they do is they transfer electrons, as you can see in the second picture, from NADPH, which is located intracellularly, to oxygen at the outer side of the membrane, and in this way superoxide is produced. If we then look to plants, they have some additional sources of reactive oxygen species, because plants are sessile organisms, cannot cope, so they will capture oxygen everywhere and they have to deal with it. In chloroplasts, we find the main production of, for instance, singlet oxygen, because chloroplasts capture the light from the sun, the energy from the sun, to give it to chlorophyll, and if this energy is transferred to oxygen, we get a production of singlet oxygen. In all the other places, we can see the production of the reactive oxygen species through electron transfer. In the chloroplasts, it's about the Maillard reaction. In the peroxisomes, as a result of photorespiration. And of course, plants also have a cell wall and cell wall peroxidases, which are linked to the cell wall formation. There we can have also hydrogen peroxide production. But what is the result of a disturbance or not of this redox balance is that we will end up with damage when we have a lot of oxidative stress. This will induce cell damage and cell death. Of course, we can have also signaling with lower amounts of redox imbalance, and this can steer the processes and are very important in metabolic reactions. If we look at the potential places where oxidative damage can occur, then we can see here our lipid membranes, in which the lipids have very long fatty acid tails, and these are prone to oxidative damage. For instance, how can it start? If we have a free hydroxyl radical, it can abstract a hydrogen atom from this fatty acid tail and as such create a lipid radical. This lipid radical can, with addition of oxygen, lead to a lipid peroxiradical, and if there are redox active elements present, like for instance copper or iron, this can eventually increase the production of lipid peroxyradicals, and finally we can have the termination where you have degradation products of these lipid radicals. If we look at potential damage at the level of the proteins, we have to make a distinction between reversible and irreversible protein oxidation. As you can see on the right hand side, we have the reactive oxygen species and nitrogen species that can attack native proteins, but the reactions on top are the ones that can be reversed with enzymatic systems, or the ones at the bottom will lead to irreversible protein oxidation. And then at the level of our DNA, oxidative stress can perform lots of problems. Let's say the hydroxylation of our guanosine base, which is mentioned completely at the left on the top. We can have inter-crosslink production or interstrand crosslink production. And these different mechanisms can all lead to different problems, outcomes that we can see at the bottom, from necrosis to mitochondrial dysfunction. Besides the oxidative damage, we also have oxidative signaling. And this signaling really depends on the ROS, which are central in this system. And they come from where are ROS produced, how can ROS be transported, are they transported, and how much can be scavenged. This all together will lead to the formation of a certain amount of reactive oxygen species that is deciphered in ROS gradients. So how much do we have? ROS signatures, which type of ROS do we have? And ROS propagation, which indicates how ROS can move via production, scavenging and transport within the system, and this all together integrates in the ROS sensing and redox regulation. This will coordinately change in physiological activities, the metabolome, proteome, methylome and transcriptome, and that all together will give rise to a response. If we look at the oxidative signaling and the half-life of ROS, which will indicate whether they can have time to migrate throughout the cell, then we can see that hydrogen peroxide is an excellent signaling molecule due to its relatively long half-life of 1 millisecond, relatively large migration distance, and it has the easy ability to cross biological membranes via aquaporins. And if we then look here, it's a specific overview of how oxidative signaling can occur in plants. We see the cell wall at the left-hand side, the plasma membrane, and then intracellular. And we can really see that oxidative stress signaling already occurs extrinsic, so it means out of the plant cell itself. Within this cell wall, we have a lot of different processes that can produce reactive oxygen species and also start the regulation of oxidative signaling. Then, once it enters into the cell, we have the intrinsic ROS signaling at the level of, for instance, kinases, but also transcription factors that really alert and really start or regulate responses within the nucleus. And also at the level of the organelles, we can have this ROS signaling, which makes an interplay also with the intracellular ROS signaling and communication via retrograde and anterograde signaling with the nucleus. Of course, what we often see is that the ROS signaling is not on its own. It is often integrated with other signaling molecules. For instance, everything related to calcium and also hormones. So you see, centrally you have the ROS with their specific signature and timing, but also they make a great interplay with the transcription networks and gene regulation in interplay with calcium, with kinases, with hormones, and so on. Specifically for oxidative signaling, hydrogen peroxide is a very important one, as a lot of hydrogen peroxide responsive genes can be monitored. So we have transcription factors that are highly sensitive to hydrogen peroxide, but also some kinases can be oxidized and in this way become active. So ROS can regulate a tremendous amount of cellular processes, the normal metabolism, for instance, epigenetics, transcription, protein homeostasis, but also in the proliferation and differentiation of cells and the process of cell death. So it really affects all biological levels we can encounter in plant cells. And what is also a very important role of this reactive oxygen species is in our immunity, with the respiratory burst, which is located in macrophages, for instance, where we try to produce extracellular reactive oxygen species in which we can then trap our pathogens and in this way destroy them. But it all comes back to the tight coordination of ROS levels to allow oxidative signaling and prevent damage. So as mentioned on top, we have the stress factors, we get the production of reactive oxygen species, also some hormones and other signals are important, and they will activate the antioxidants so that we get a concentration gradient in which with modest elevated ROS levels we still, or an organism can still adapt to the stress. Most often this goes hand in hand with an impaired growth and development, but if the stress becomes too high, we will get programmed cell death and necrosis. Which are now these antioxidants? They consist of enzymes on one hand side and metabolites on the other hand side. Enzymes that are present in all organisms, metabolites that we often have to consume through plants that are able to produce these metabolites, but we have lost these biosynthetic pathways. If you look into the enzymes, we have superoxide dismutase that can convert the highly reactive superoxide to hydrogen peroxide and molecular oxygen. And we can distinct the different superoxide dismutases by their dependence on the metal cofactor. To dismutate hydrogen peroxide or to scavenge hydrogen peroxide. Catalase and peroxidases are very important. Catalase can work with hydrogen peroxide as the solely substrate, while peroxidases need an organic electron donor to be able to convert hydrogen peroxide to water. And then often there is an interplay between the metabolites and the enzymes, and a good example is here in plants, where ascorbate and glutathione as metabolites work together with different enzymes to detoxify hydrogen peroxide to water with the use of enzymes and metabolites in redox cycling. Then we have the peroxiredoxin thioredoxin system, also important to convert hydrogen peroxide to water with the interplay of metabolites and enzymes. And what we can see here at the end is that we use electrons from NADPH to help this process. But in both cases, the ascorbate glutathione pathway and the peroxiredoxin signaling cycling, there is a central role for glutathione, as you can see here. But the regeneration of glutathione also relies always on the NADPH as an electron donor. That is really important to help this process. So, in summary, the Ross signature, which is here envisioned in the middle of the figure, is very important to steer processes. And this Ross signature is a balance between the Ross itself and the antioxidant defense systems. And the Ross are derived from endogenous sources on one hand side by normal activities of our organelles, but also from exogenous sources like, for instance, these abiotic stress factors. And this redox balance will really determine the outcome of it being a homeostasis, so we need the redox balance to have the normal growth and metabolism ongoing. But when it's reduced function, then we can have defects in host defense responses and also in cell proliferation. And on the other hand, if we have too much oxidative stress, then we can have random cellular damage, but also specific signaling pathways that will give rise to disease, aging, apoptosis, so a negative outcome for the organisms. So, maintaining a good and proper redox balance is essential for normal life on Earth.